Hello and welcome to AI4. Our next session will be led by Max Bennett. He is the co-founder and chief product officer at Bluecore. Please join me in welcoming Max to the virtual stage.
Hey folks, thank you. First of all, thank you to the whole AI4 team for putting on this awesome event. I'm super pumped to be here. So can you see my screen? Cool. Okay, so I'm the co-founder and chief product officer at Bluecore. We're a software company, mostly in New York, that applies machine learning to help retailers better personalize marketing. I'm not going to talk at all about that today. What I'm going to talk about is a passion project that's been something I've been thinking about a lot and working on over the last few years. And that's been trying to understand and decode how intelligence is implemented in the brain. This is something that I've been very, very fascinated by. So hopefully some of this will be interesting and applicable to folks, specifically those who are interested in machine learning and how to improve AI. So the first very legitimate question to ask is, why should we look into the brain at all? We've seen incredible success over the last 10 to 15 years in applying the existing machine learning techniques, and without obsessing over what works in the brain, we've been able to do things like Alexa. We have incredible natural language processing recommendations. We can diagnose medical images. We can do facial recognition, self-driving cars. We can beat humans in AlphaGo. So is the brain even important? And I want to argue to you that the brain is very important for two reasons. One, much of the success that we've seen in AI over the last 20 years actually has come from looking into the brain in the first place. So our model neuron that we use in all of our neural nets is devised from our understanding of what neurons were doing. This was our understanding 70 years ago. But the model neuron is very similar to that Frank Rosenblatt came up with in 1958. I mean, he came up with this idea by understanding how neurons actually work. So that's one reason, which is the brain has already given us a lot of insight. But the second reason is there's still a lot of stuff we can't do. So if we look to sci-fi movies and books, there's still so many really cool things that we kind of imagine doing that we don't know how to implement yet. So, you know, from the movie Prometheus, there's these robots that can diagnose disease and cure you, and just by asking you questions and scanning your body, or, you know, robot and Frank, there's movies where there's robots that take care of the elderly and help them feel warm and appreciated and build friendships with them. There's teachers that can tailor education to each individual student. There's assistants that can do dishes and can help us around the house or protect us. And there's all these amazing things that we imagine AI being able to do that we still don't understand how to do. And what's interesting is all of these things are things that the human brain does. So all of these great ideas are really just trying to figure out how can we implement what the brain does in an algorithm. And put maybe slightly more formally, there's a bunch of things that the human brain does that blows all status quo AI out of the water. One is online learning. The brain is way better at learning as it goes. The brain is way better than any state-of-the-art technique when it comes to generalization and being flexible. The brain can learn lots of different things. The brain is way better at one-shot learning, meaning not getting a bunch of training examples in order to understand something, but it can experience something once and immediately generalize and apply it. The brain is way better at sensorimotor learning. We still don't have robots that can just do the dishes. That is an unbelievably complex robotics task that humans do effortlessly. And then lastly, the brain runs on the wattage of a light bulb. It's unbelievably efficient. So there's a lot we can learn from the brain, but it's really hard. So one reason why people avoid doing this is because it's really challenging. The brain has over 100 trillion synapses, 85 billion neurons. All of them are really, really small and compact. And even just the connections dramatically understate how complex the brain is. There's all these neuromodulators or chemicals that when they're present completely change the learning rules and the computations and all of that stuff. But if we try and break down the problem a little bit, you can kind of break it down into two pieces. One is just understanding what does an individual neuron do? And understanding what are the computations performed by one neuron. And then secondarily, if we understand that, then we just have to look at the actual circuits that are being implemented and try and decode what are the computations such circuits are doing. Okay, so I want to talk about each of these in sequence. So first I'm going to talk about understanding the computations of a single neuron. So you're probably very familiar with the standard model of neurons. Now this is a model of neurons that was devised over 70 years ago based on our understanding of how neurons worked at the time. And this is exactly what we still do in neural nets today. There's an input that comes from some other neuron with some real valued, some scalar value. It multiplies by some weight. And then there's maybe some sigmoid function that sums up some linear combination of these inputs and then takes a uniform output and passes to the neuron it connects to. Now there's components of this that are accurate. There are weights, and these weights change with learning. We see that in the brain. There is uniform output that neurons send. They don't send different output to different places. But there's a lot about this standard model that we know now is wrong. And neuroscientists have made so much progress over the last 70 years understanding what are these individual neurons actually doing. And unless we sort of augment our standard model of neurons, it's going to be very, very hard to decode circuits that are leveraging computations the standard model does not incorporate. So I think there's many, but I think there's three really key ways we need to update our standard neuron model. And I want to walk through each of them and sort of try to give some intuition onto them. So the first one is spiking, the incorporation of timing. So if you were to go into a brain and you put a little microelectrode on any one of the neurons, and a microelectrode is just trying to measure voltage, but you think about it as charge. What you would see is you'd see these spikes. You would see normally a neuron is at some resting potential at about -70 millivolts, and spontaneously for really only a millisecond or so, it will rapidly go to 30 millivolts and then come right back down. So what's interesting about this is the brain isn't passing the scalar value. It's not fluctuating from -70 millivolts to 0 to 30 back down. It seems to be in some ways analogous to binary in a computer in some ways, where it's either on or off. Now, some people have tried to interpret these spikes as, well, maybe we can think about it just as a rate. So if we kind of create a bucket, we can say, well, this is maybe 3 because there's 3 spikes in this time period. And then this maybe goes to 7, and then it goes down to 0, and then this goes to 4. And we can try and interpret spikes by coding them into rates, meaning what's the frequency of firing. And that's called the rate coding hypothesis. There's been a lot of research that shows that the rate coding hypothesis is, at least in several important ways, incorrect. And I want to show you some examples. So if you zoom into what's happening in an individual neuron, what you see is something that looks similar to our model of neuron. So if A, which is an axon from another neuron that attaches the cell body of our neuron, fires, what you'll see is this -70 millivolts will increase, not enough to a threshold. So spiking only occurs if a threshold is achieved, and then it will decay back down. So if these two are separated far enough in time, it won't really reach the threshold. But if you fire A rapidly enough, in temporal summation, meaning in rapid enough succession, this increase might happen. It might actually reach the point where a threshold, then you might get a spike. Similarly, there's spatial summation, meaning multiple axons can fire, or these synapses can, presynaptic synapses can fire at the same time, and then you also get a summation. So this is just an example of what you see start to happening. In this specific example, it's only going to fire when A happens rapidly, and then B happens rapidly, and then you get a spike. Okay, and then there's also, of course, this inhibitory neurons that attach. Okay, so does this matter? Is this really that different than our model of neuron? Well, let me show you an example where it is. So let's say, basic example, we have one neuron with 3 inputs. And let's say I give 10 hertz, which is frequency of spikes, through the top one, 5 hertz at the bottom one, and then 10 hertz at the bottom one, 5 hertz in the middle, sorry. And the bottom one is going to be inhibitory, meaning that when this fires, it actually lowers the potential. It subtracts against the other ones. So what you might see here, if the weights are all the same, is that this 10 hertz of inhibition kind of cancels perfectly the 10 hertz of excitation, and then you kind of get 5 hertz that just kind of flows through. So this works exactly the way our standard model works. But if I made one tiny modification, the outcome completely changes. What if I just phase shift this frequency back 2 milliseconds? If I just move it back slightly, it completely changes the outcome, because timing matters, because this neuron fires first when it gets this spike before any of these other effects actually take place. So trying to understand things just in rates misses some of the really key things that neurons are doing, where timing really matters. When things arrive are encoding some form of information. I'll give you another example where this happens all the time in the brain, and we think these are really common circuits, something called a winner-take-all network. So imagine you have a network where there's 3 neurons, and they all get the same input. So let's say just 4 hertz. You have 4 spikes that each of them get. And let's say it takes 4 spikes for these neurons to fire a spike. So when each of them gets these 4 neurons, it'll spike. They also synapse on this inhibitory neuron, which then inhibits all of them. Okay, so not that interesting, right? It gets 4 spikes, it fires, then it inhibits this, and then all of them get inhibited. But if I change the timing slightly, if I phase shifted the input to these 2 back just a few milliseconds, meaning the top neuron fires first, all of a sudden you get a completely different result. If this neuron fires a little bit before them, then by the time this neuron was about to get enough summation to fire, it's already being inhibited by this neuron. So we see networks like this a lot in the brain, where it uses timing to completely change the outcome of networks. Okay, so hopefully that gives you a little bit of intuition. I'm gonna obviously go into more detail later on applying this type of augmented neural processing, but hopefully that gives you some intuition as to why timing matters. The other update is dendritic integration. So let me quickly explain that. So if you look at a neuron, you see these huge like spikes coming out of them. These are all dendrites, and dendrites are just where other neurons synapse onto a neuron. Think about them as places or points of input. And you can kind of classify the synapses that attach to a neuron into 2 buckets. You can, there can be the proximal synapses that attach really, really close to the cell body or soma. And then there are the synapses that attach really, really far away on these distant or distal dendrites. And the majority of synapses on a neuron in the brain actually attach on these distal dendrites. Okay, so does it matter? Who cares? Well, if you record the input from an axon to one of these on a proximal synapse, you see something that you would expect. If there's an action potential on the input to this neuron, you see some increase in voltage, right? So this is just summing at the soma. This is exactly our standard model. But if I do the same thing in any one of these synapses that are distant from the soma, you see no effect, 0. So when any one of these spikes, you don't see a little bit of summation, you see no effect at the soma. So what this would imply is that these distal synapses do nothing. It would imply that 70% of the input to neurons in the brain have no effect, which seems counterintuitive. If they don't add up, then they're not having any effect on computation. So let's dive into this a little bit more. Instead of recording the soma, let's record the actual dendrite. And here you start seeing something interesting. If I have a spike on one of the neurons attaching to this dendrite, you will see an increase in voltage. But what happens is by the time that propagates down all the way to the cell body, it's become nothing. It becomes 0. So now if you keep adding more input to a dendrite, you see something that only really has been better understood in the last 10 to 20 years, which is a dendritic spike. So if there's enough summation at the dendrite, then the dendrite itself spikes. And that spike will propagate all the way down to the soma, and then you get a little bit of summation. Now, it's not a whole spike, but now you're actually adding at the soma. So what this means is, if we observe a neuron that's getting input at the cell body and then input at its dendrites, the right way to model this and think about it is not as a neuron that just has a bunch of input, which is the standard way of doing this, but instead it's to understand that there's at least two integration points. First, there is summation at the dendrite, and then the output of a spike from a dendrite is summed at the soma, and that's the output of a neuron. So, okay, so does this really matter? I'll give you one example where this is really important. So let's go back to our winner-take-all network. And let's assume that the first neuron has a distal dendrite that's getting input from these 3 other neurons. Now, the first thing you'll realize is that these are very noise resilient. They actually, they filter out noise. And the way they do that is if you had all of these inputs that were attaching at the soma and you had thousands of synapses, eventually just by random spikes, it would start changing the outcome of this network. But with these distal dendrites, it has no effect on the soma unless there's a spike. So it filters out all the noise. That's one really good thing. Secondarily, and perhaps more importantly, if there is a spike, it can change the outcome of winner-take-all networks. So let's say this neuron has a dendritic spike. That means even though the input to all three neurons is exactly the same, the first neuron will spike first because it actually got more input from the dendrite. And that will completely change the outcome of the winner-take-all network. So what we start to see is these winner-take-all networks incorporate timing into their computation, as well as on all of their hundreds of dendrites. If any of the dendrites are receiving a pattern or spike, it can completely change the outcome of these networks. Okay, so that's hopefully gives a little bit of intuition around dendritic integration. And then the last really key thing is local synaptic learning. So we're probably very familiar with the idea of backpropagation. It is extremely successful. We use it all the time. The problem is the vast majority of neuroscientists, not all, but the vast majority of neuroscientists agree that this is completely biologically implausible. As a quick example, if backprop works in the brain, that means that when there is an error, when there's something supervised, and I compute the error, I need to update the weight over here based on exactly what the partial derivative is of changing this weight relative to this error. So I need to propagate back the error all the way back to this neuron. So there are very, very smart people trying to figure out how this might happen. Jeffrey Hinton is working on it. So it's possible they uncover how this might happen. But most neuroscientists agree that it doesn't seem like the brain is really doing this. And the type of learning that we know for a fact happens in the brain are these local timing rules at synapses. So I think it behooves us to try and model these things that we know happen as opposed to things that we think should happen. So I'll give you an example of one type of learning rule we see everywhere in the brain, or in lots of places in the brain, and something called spike timing dependent plasticity. What this means is if you look at a synapse, so a connection between one neuron and another, and you record each of these, if there is a spike on the presynaptic, meaning the input neuron, before the output neuron fires, so you see this spike and then this spike, meaning that there's some implication of cause. The input neuron seems to, in some ways, maybe have caused the output neuron. You see the weight increase. And this can happen in one shot. It can automatically increase the weight of that synapse. On the flip side, if there's negative timing, meaning that if this input neuron fires after the output neuron, meaning it doesn't seem to have caused anything, you see the synaptic weight decrease. So there's all these really interesting timing rules. It seems like perhaps this is trying to be a causality detector. It's only increasing weights when things cause each other and decreasing them when they don't. Okay, so there's at least three key ways I think we want to update our understanding of neurons to decode the circuits we observe in the brain. One is shifting away from rates and thinking about spikes and timing. The other is making sure we're incorporating multiple steps in integration in our model neurons. And the last is trying to incorporate timing rules. There's lots of different timing rules. These timing rules can change at the same synapse based on things like neuromodulators. It gets very complicated. But the point is, we probably want to model at least some timing rules. Okay. So this gives us an understanding of the computations that are happening in a single neuron. And I'd say, we're still learning a lot, for sure, but I'd say we're like 75% as a neuroscience community at understanding what's actually happening in neuron. Where we have a lot to go, where I'd say we're at 15% of the way there, is understanding what are the actual circuits implemented in the brain. So now I want to sort of shift gears towards, okay, let's apply what we think we understand about how neurons are computing things and transforming input to decipher what circuits are happening in the brain. Okay. Now, the really hard part about this is there are so many different subcomponents in the human brain. And each of these subcomponents have millions to billions of neurons, billions of synapses. They have completely different circuitry. They're all highly interconnected with each other. So it's very, very hard to decide where to start or even how to decipher this. Now, there's a lot of excitement in one particular region, which is the one I want to talk about, and it's called the neocortex. So a lot of people think if we want to start learning more about how the brain implements intelligence-like things, the neocortex is a good place to start. And the reason is because this neocortex seems to be where all the smart stuff happens in the brain. It seems to be where object recognition happens, reasoning. It seems to be where we learn motor tasks, make decisions, It's where language seems to be, working memory, facial recognition, music perception, all these really special things seem to happen there. Also, interestingly, the most intelligent animals are all mammals. They're monkeys, dolphins, humans, and the only subset of species that even have a neocortex are mammals. So it seems to be the case that neocortex is related evolutionarily to some step function forward in intelligent function. Making this even more sort of salient, there's a lot of studies that show this, but just to sort of make the point go home, is if you lesion or get strokes or damage to these different parts of your neocortex, you completely lose certain abilities. I mean, if you get a lesion in the back part of your neocortex, you can become completely blind. If you get a lesion somewhere here on your temporal cortex, you can forget faces, lose the ability to recognize faces or objects. You can become paralyzed in certain places or lose the memory of how to do complex skilled things like walking or playing music. Language is a specific part of the brain that if it gets damaged, you lose the ability to understand language. So it seems to be necessary to a lot of these smart things. So let's try and sort of decipher what circuits are actually happening here. So if you take the brain and you created a slice through it, and you just looked at a slice through the brain, the neocortex is this sort of film that goes all the way around and wraps around this inner part of the brain. Okay, and it goes all the way around. So if we zoom in a little bit, and we took a little slice out, it seems like there's these 6 layers of neurons that are all compactly pressed, and they wrap all the way around. Now, what's interesting is the neocortex clearly does all these different things. It's a region that processes images, sounds, language, motor learning, all this fun stuff. But if you start looking at these little cutouts of the neocortex all over, in almost all places of the neocortex, the neurons, the structure, everything seems exactly the same. Even though the neocortex is doing all these crazy different things, it seems like they're all exactly the same. And it seems to be that there's these little 500 micron wide columns, sort of a section through this surface where most of the connectivity, not all, but most of the connectivity is all local to this little circuit. This is really interesting. How can this sort of uniform thing be doing such a broad set of intelligent-like functions? And this idea that Vernon Mountcastle came up with in the 70s, which is kind of mind-blowing. It's this idea that actually the neocortex is about a 1 to 5 million repetition of the exact same little microcircuit. It's only about 500 microns wide. And the only difference between any 2 regions is the inputs it receives. Now, if this is true, this is extremely exciting because it simplifies the challenge of saying, how do I understand the circuit implemented by the trillions of synapses in the cortex, to just trying to understand this one little microcircuit that's being replicated a few million times. Okay, so the huge question is, what are the computations implemented by this neocortical microcircuit, assuming there is one. There's lots of evidence to suggest there is. And this is so exciting because if one circuit can do such a broad set of intelligent tasks, maybe it can garner some insights into us onto how one-shot learning happens, and how online learning happens in the brain, how generalization occurs. There's all these really interesting things we think we could learn. So there's a ton of crazy smart people that have been working on this for a long time. Here's just a sampling of those folks. These folks are some heroes of mine. And all of them have been making incredible progress into our theoretical understanding as to what this microcircuit's doing. Okay, and we're sort of entering this golden era of understanding what the microcircuit does. But we haven't yet figured it out. And my benchmark for figuring it out is, can we implement something that does all of these special things that the brain does in a computer? And if we can't, then we don't really understand exactly what's happening. And I think we haven't figured it out for a few reasons. The obvious one is that neurobiologists are still uncovering what the actual circuits and connectivity is within the microcircuit. So we don't have full information there. But even in the last 5 to 10 years, we've learned a ton. But the other ones are more in our control. Not all, but many theorists who are trying to interpret what's happening in the microcircuit still use this old model of neurons from 70 years ago. We're not using spiking networks. We're not using timing. We're not using dendritic integration. And that's a problem because it's going to make it hard to interpret these circuits that we think are using these types of computations. And the last one is we don't use continuous time. So we don't use, the brain doesn't have a training set and then goes live. The brain, from the moment it's born, is getting a continuous stream of spiking data from your sensory organs all the way until the point that it dies. There's no real on or off. You could argue in some sense sleep is off, but really the brain is learning in this continuous fashion. So we need to incorporate that, I think, into understanding what the circuits are doing. So I recently, actually just last month, published a paper where I'm trying to sort of understand some of these things. And what I do is I incorporate up-to-date observed circuitry with up-to-date understanding of neurons in continuous time, and try to unify a lot of these ideas from these other really amazing thinkers into trying to have one sort of unified theory as to what it does. And it proposes a potential answer to how one circuit can do all these things. So what I want to sort of walk you through with the second half of this presentation now is, what is this circuit doing? Okay, so here's kind of what I think is happening, which is consistent with what other folks have proposed. I think the way that the brain is able to, this microcircuit can learn visual things and auditory things and somatosensory things, et cetera, is it's just a generic prediction and sequence clustering machine with some special features. It receives input spikes and it learns to predict what spikes it thinks it will receive next. And it clusters common spikes that it gets and patterns it gets into a sequence code. So it does some dimensionality reduction or some unsupervised learning on its inputs. So to understand that, if we zoom in on the circuit, there's 6 main types of neurons. You don't need to sort of remember this or anything, but just know that there's 6 main classifications of excitatory neurons and what we see commonly in the microcircuit. And what I'm going to do to try to elucidate our understanding of it is I'm going to use this kind of visual model or example of what's happening. So each one of these circles represents a neuron or where the soma is, even though its circuitry and dendrites might be all over the place. And the number of neurons in a given macro column is way more than this. So it's, I'm simplifying, but the idea is to try and drive our intuition. So I'm going to use this to try and explain things. And neurobiologists have explained a lot of the circuitry that occurs. So again, you don't have to memorize this, but what I've tried to do is sort of bring together our understanding of the mapping between these different neurons in other parts of the brain and each other. Okay, so let's follow the circuitry. So input into this microcircuit all generally goes into this layer 4 set of neurons. And what we think is happening here is coincidence detection. Now, most people would generally agree with this. And there's a lot of evidence for this. If I look at the neurons that are input to just the visual cortex, so the part of the brain that receives input from my eyes, you'll detect in this part of the brain called the LGN, don't worry about that, but I'll detect neurons that respond to specific circles of light and places in my field of view. So maybe one neuron always fires when there's a light in the top right corner, there's one that's a little bit over here. But if you record the neurons in layer 4, what you see is actually things that respond, neurons that respond to bars of light of specific orientations. And if you think about that a little bit, it's almost obvious that, well, a bar of light of a specific orientation is just a coincidence. It's a summation of circles or dots in a specific orientation. So there's a lot of evidence that what's happening is it's just summing coincidences, which is straightforward on a neuron. So in our conceptual model, objects or coincidences or patterns are recognized, are sort of represented by a specific activation of a pattern of L4, these blue neurons. And what we know is these L4 neurons fire an entire mini column when they get activated. So these little mini column representations are representations of objects. And this is very commonly how other people have been sort of modeling what the macro column does. Okay. But the real question for us is the thing that the human brain seems to be doing that's really interesting is we seem to predict forward in time. So when I show you this image, you can, without thinking at all, you can start predicting forward what's going to happen next. It almost looks like it's in motion. You can see that her leg is going to come down and kick this ball. And so how's your brain predicting forward in time? So as another example here, I'm going to play a 3-note sequence. It's not going to be magical music, but I'm just going to play a 3-note sequence. And then I'm going to play the first 2 notes. And in your head, you're going to easily be able to hear the 3rd note, even though I only showed this to you once. So your brain's going to do one-shot sequence learning. So let's do that. Okay. It's so trivial that it's almost not even interesting, which is, of course, you hear that 3rd note in your head. But it's actually kind of magical. Your brain one-shot learned a sequence. Okay, let's do something a little bit harder. What if I input into your brain 2 very overlapping sequences? You don't have to understand the music here, but just know that the middle 3 notes are identical. The only difference in these two sequences are the first and last note. So I'm gonna one shot, I'm gonna play sequence 1, I'm gonna play sequence 2, then I'm gonna play this, which is one of the two, I'm not gonna tell you which one, and I'm gonna omit the last note. And you're gonna tell me if your brain can easily disambiguate what the last note is. Is it a high note or is it a low note? And if your brain's able to do that, it somehow robustly maintained a sequence, even though for the 3 last notes, it was identical. It somehow maintained it from the first note. So let's try that. Okay, high note. Okay. So almost everyone universally hears it goes to the high note. But that's actually kind of crazy that your brain just did that. I only played these sequences a single time, and yet it was easily able to disambiguate these sequences, even though they're mostly overlapping and identical. Okay, so how is the brain doing this? How is it one-shot learning sequences, disambiguating overlapping sequences, and predicting the next element in a sequence? And maybe if we understand this, it can help us understand how one circuit's doing such a broad set of things. Okay, so let's sort of play out the circuitry. So if we input a sequence ABC, and let's assume we input it really rapidly, so A, then B, then C really quickly. Let's see what would happen. So we know that these layer 2, 3 neurons, these green neurons, project to these other set of neurons in layer 5A. You don't have to really understand that, but the point is, we're pretty sure that it does pattern separation. What that means is a random subset of these layer 5A neurons will fire depending on what's firing in the green neurons. And we also know that these layer 5 neurons project back onto these layer 2, 3 neurons, the green neurons. And we know what they do is they're kind of sparse. They target random neurons. It's not related to all of them, it's just to some of them. So what this means is a subset of neurons here will get a little bit of a depolarization or increase in voltage. Now, if I activate B before that decays, and we know that these green neurons implement a winner-take-all network, which we see regularly, you're now going to get a different pattern. It's going to change the outcome of the winner-take-all network. You're going to get the columns for B, but it's going to be sparse. Some of them won't be active. It's only going to be the ones that were activated by this. So that now means that this represents a unique code for A to B. And if you follow the same thing at the end of the sequence ABC, you get this unique code just for the sequence ABC. So this sort of progressive sparsity is this brilliant idea that Jeff Hawkins came up with. He thinks it also happens in layer 2, 3. I propose a slightly different circuit, but the idea is something he came up with, and it's amazing. And it's really cool how, if you follow this forward, you can kind of see how maybe a macro column could learn a whole sequence. Now, with synaptic learning, when this happens, when this fires, you're going to get immediate potentiation of the weights between these layer 5A neurons and these neurons. What that means is if I put in A again, the whole thing can get replayed, because these A neurons are going to project here, which is going to then project exactly the neurons that it was potentiated, which is then going to go to another sparse representation here, which is then going to play A. And what you can show, I don't have time here to explain exactly how, but you can show that this whole sequence would also get clustered into a unique code in these layer 5B neurons. So now what you have is you have the ability to replay a sequence after hearing it once and cluster the whole sequence together into this unique sequence code. So this is super cool. And this sort of explains how your brain is able to easily tell the difference between these two sequences, even though, if it's C, B, C, D, C, even though these middle patterns between these two sequences are identical, you can see the sparse patterns are completely different because it started in a different place. Cool, so now we're starting to understand how these things are disambiguating, but I didn't explain really how prediction happens, right? So if I omit the last note, you can still kind of tell what's going to happen next. And in order for that to happen, this macro column, when it's in the D in this sequence, needs to predict C, needs to output some prediction somewhere else. Because if you're able to say what you predict, it can't just live only in the macro column, it needs to send to other subsystems. And then it somehow needs to output a prediction for A if you're in this sequence. So how does it do that? So if, again, we follow the circuitry, what I think is happening is these layer 6A neurons also get input from the thalamus. So input into a macro column not only goes to layer 4 neurons, but also goes to distal dendrites of layer 6A neurons. So what that means is any given object is represented by activation here, but by a sub-threshold depolarization or a little bit of increase in voltage here for where the dendrite spike, there's dendritic spikes here. Okay, and now we also know that these layer 5B neurons project to random subsets of these purple neurons, these layer 6A neurons. So what this means is for every sequence code, you get a random sampling of layer 6A neurons. So if you bring these two things together, you get something super interesting. What you get is a unique code for the element A in the sequence ABC. And you can show this with the winner-take-all network. So this means that these layer 6 neurons represent unique element sequence codes. So if for A and ABC, A and AZY, A and AXW, you get completely different codes here. And you can show how this enables you to map this element A to a different next element. And what's cool is you can also show, I'm slowly running out of time, so I'll move fast. You can show how these neurons project back to where neurons get input, and they project actually also to layer 4 onto distal dendrites. So it seems like they're predicting the next element, even based on their circuitry. So now what the proposal is what's actually happening is when you have C, these purple neurons predict B, and when you have B, these purple neurons predict C, so on and so forth. And you can kind of show how that might be implemented. Okay, so we now showed how these three things. So I want to sort of quickly go through some other cool things that might be in this microcircuit. The first thing I need to do is I need to answer a key flawed assumption. In order for what I just described to you to work, I needed to input ABC within milliseconds. It needed to be the case that when B showed up, a random sampling of these neurons were depolarized, means were affected by these layer 5A neurons from A spiking. But if it happened too far away, if B was input 5 seconds later, you would just get the normal representation of B. That's problematic. So the problem is the window for synaptic learning is 50 milliseconds, but clearly we can learn things when they're separated by seconds to minutes. So how do you do that? So let's walk through a learning paradigm. If I give you A, I pause for 5 seconds, I give you B, pause for 5 seconds, I give you C, you can clearly learn this sequence. So how does that happen? So what I think is happening is it takes advantage of working memory or the hippocampus. If you go into the hippocampus, you know that there's these theta oscillations. In other words, it's replaying something, and we know these to be what's sometimes called place codes or episode codes. It's unique code every 200 milliseconds. And it seems to go to this part of the brain called the ACC, and the ACC seems to project all over sensory cortex, specifically to these layer 6A neurons. The circuitry shows that. So what's cool is it seems to be the hippocampus takes a random episode code, and I don't have time to talk about exactly what that does, but just assume it's a random code for a moment in time, and keeps replaying that to the whole sensory cortex. And what you can show is how that actually enables you to connect things across time. So if this code goes to layer 6A, and then you experience something, a very new circuitry that was recently discovered, shows how these layer 6A neurons can actually reactivate an entire representation in layer 2, 3. So what that means is this episode code, if it keeps projecting a random code to layer 6A, you can reactivate memories without ever getting sensory input. So this is how I think working memory happens. Lots of evidence for this. I don't have time to go through it, but you can see that working memory activates features held in memory. You can show that working memory skips all layers except for layer 4, which is what you see in the prior slide. You can see that projections from higher order cortex go specifically to deep layers, which means probably layer 6A. And you can see that firing seems to propagate from layer 6 upwards, which is another thing that it's consistent with the model or project. So what you can show is as long as the hippocampus is replaying this episode code consistently, it can be random as long as it's consistent, all of a sudden you can easily connect ABC together even though they're separated in time. And the intuition is even though B is separated by 5 seconds by A, if the hippocampus is replaying the experience of A, then on the synaptic level, it actually was in milliseconds apart because it keeps replaying. Okay, I also think this is how episodic memory might be happening. Now, because a memory is a distributed representation probably across the back of your neocortex. So any random episode code can get mapped to an entire representation across all these different macro columns. This also explains why if you get damage to your hippocampus, you can completely lose the ability to generate new memories. Okay, this explains working memory, warning over delay periods. One of the last problems that we need to solve is something called the stability plasticity dilemma, something that Stephen Grossberg talked about in the 80s. And what this represents is the problem that if you have an online learning system and you're constantly streaming information, it risks catastrophic forgetting. If I'm constantly updating weight space on what I'm seeing, how do I make sure that my prior representations maintain robustness and I don't risk overgeneralizing and all of that? So he came up with a really interesting idea that this happens in the thalamus, and I think he's right. So if you track what a macro column does, it receives input from the thalamus, which is where it gets sensory input, goes to this part of the brain called the thalamus, to layer 4. And then you get this whole prediction that goes back to the thalamus. So this prediction seems to go back to where it gets input. And if you track the circuitry, you don't have to understand this, but the main output is that it seems to do different things when prediction is correct versus when prediction is not correct. The circuitry seems to compute what the macro column thought was going to happen is that actually what happened. And what you can show is when it's wrong, when there's a mismatch, how that mismatch actually wipes representations in the macro column. So what effectively we think is happening is new learning only happens to solve mismatches. And that helps solve the stability plasticity dilemma, which is the thalamus is only firing a specific type of neuron if a macro column fails. And if a macro column fails, that's when a macro column starts going through plasticity processes to learn new things. So unless predictions are false, the brain doesn't need to learn new things. And that's one way the brain might be solving this online learning problem. Okay, so last thing, sensorimotor prediction. So as you're running down the sidewalk, you're getting a stream of sensory information coming towards you. And that is not alarming. But if you were standing and not moving, and that same thing happened, all of a sudden things move alerts towards you, that would be extremely alarming. Somehow the brain tells the difference between you moving or the environment moving. Another example is if you look around this room, I show you this room, you look at the desk, you look outside, the painting, the chair, you don't think it's moving, but what's actually going into your eyeball is this shift in a picture. So somehow the brain tells that what just happened now is the picture moving, but when your eye did it, it was your eye moving. So how does the brain sort of predict expected outcomes of sensory movement, saying, am I moving or is the object moving? So here's how I think that happens. Imagine there's something in your left, let's say there's a bar in your left receptive field and you want to look over to see it. So one macro column gets currently already is getting input from this, so it represents this bar. And the center one that's about to see it, meaning you're about to look to the left and it's going to be in its field of vision, has no input. Now what we can see is these layer 5B neurons project to these other types of neurons, these yellow neurons, cortical neurons. And what I think happens is motor commands also seem to project to these. There's other sort of structures that would explain the same thing, but this is a simple proposal or way it might work. So now what happens is this code here represents a specific code for a 45-degree bar here, plus the motor command to look to the left, and that projects over here. Okay, so now you get a unique code, which is in the center macro column. These cortical neurons project across columns. You get this unique code. And now if I see a left bar here, all of a sudden what happens is you created a mapping between left bar over here, the desire to move your eye to the left, and center bar here. You created a mapping between movement and sensory experience. So what does this mean? This means that the next time you have this exact same situation and you want to look to the left, you're going to predict that there's going to be a 45-degree bar in the center view before it happens, because this mapping will occur where these layer 6A neurons, these purple neurons, will predict it before it happens. And this is really interesting. This might explain some of the problems we have in vision, which is how does the brain do such a good job of generalizing to orientation changes and all that fun stuff. Okay, and then of course the brain seems to stack these macro columns on top of each other and create hierarchies, and you can create really compound abstractions and sequences of sequences and sequences and all these really interesting things. Okay, so I know that was a lot and I'm probably way over time, but there's lots more work to do. The model I proposed is highly falsifiable. So I listed a bunch of sort of tests you can do to evaluate its truth. There's lots of other people that have come up with very legitimate alternative models that also are falsifiable. So there's a lot of work to be done on experimentation to see the difference. And of course, the real test is implementing these things in actual algorithms and computers to see if they do anything interesting. So this is sort of one next step. And then from a neuroscience community, there's lots more to understand about neurons and brain circuits. So So my hope is in this sort of symbiosis between neuroscience and AI, where neuroscience is answering how the brain does stuff, and AI talks about what happens in practice. My hope is sort of this type of thinking can give ideas on how to do things to the AI community and eventually get to better AI technology. So it's my talk. I'm happy to answer any questions if there's any time for it.
Awesome. Thank you so much, Max. Now I have that tune stuck in my head, so I guess that means my brain is learning. All right, thank you. And feel free to drop your questions in the chat so that Max can interact with you there. If you're looking for your next session, take the time to check out the program page and find where you're headed next. Also, it's a great time to start some conversations with your peers with some virtual networking. Thank you again, and we'll see you around.